Hey guys, welcome to AP Biology or College Biology. We're going to be focusing in on Unit 5, Heredity, Section 5.4, which is all about non-Mendelian genetics. For this section, we're not going to be doing one quiz, but we're going to be focusing on several types of problems and the way that you answer these types of problems as it relates to non-Mendelian genetics. So if you're looking for sample problems, this is a great video for you. But before we get started, please note the disclaimer listed below. Okay, let's talk about multiple alleles. So, in humans, we believe that specifically when we're talking about blood types, we note that there are different types of blood types. And this is interesting because blood types are a example of multiple alleles. And the reason why they're a great example of multiple alleles is because you don't just have a dominant and recessive form of an allele. Like if you remember with Mendelian genetics, for example, if we were looking at P height, we would have a dominant and recessive allele. And the dominant would tend to take on the properties of the allele phenotype. So what I mean by that is, say for example, we say capital A equals tall, and then we have lowercase a that equals short. We noted that if there was a heterozygous allele combination with big A, little a, in the way that we talked about it for section 5.3, we noted that the capital A would be taking over. So phenotypically, the individual would look tall versus short. It's sort of like the little a is ignored. Well, with multiple alleles, what happens is you can get more than two versions of a gene, and heterozygous tends to give a very unique combination. So, for example, if we take a look at blood types are an example of multiple alleles. We have a blood type, which genotypically can be seen as two capital A's. We designate this I indication just as a placeholder, just to note that this is blood type. So for genotype for blood type A, yeah, you could have two dominant capital A's for blood type A for the allele combination, or you could just have one capital A. And so they have a lowercase I just as a placeholder to designate those alleles. The same follows through with the genotype for B blood type. So, for example, you'll have two capital B's with that designated holder of the capital I, and that designates blood type B. Or you could have the capital B and the placeholder of the lowercase I as your blood type B. Now, if you have a combination of both dominant alleles, A and B, you get another unique blood type, which is AB. So you could see here that there are multiple capital letters for blood type designation, and the combination of both dominant alleles gives a whole new blood type. And then finally, you have blood type O, which is the act of not having any allele association, but we represent it with two lowercase i's. In this case, type O, blood type, by the way, is universal donor because if you notice here, they don't have any allele indication attached to that I. And the universal receiver, a blood type, is AB because they can receive blood type A and be fine because they already have A. They can receive blood type B and be fine, or they can receive O and it's just the absence. So you can think of the way to think of multiple alleles as what has and what hasn't. Okay, so we're going to take a look at a practice problem. So you can see here it says imagine two individuals and parent one and parent two are planning to have a child. Parent one has blood type AB and parent two has blood type O. What is the possible blood types for each offspring and their possibilities of each? So I want you to take a minute, pause this video and try to do this practice problem just like you would for a quiz question that we would have posted here. And then we're going to review that in just a second. Okay, now that you're done reviewing this problem, let's take a look at the answer together. So, when we're talking about blood type and we're taking a look at the designation here, we have the one parent that has blood type A and B. I should choose a different color here, so I'm going to choose a green. So, that's the first parent. And then in the second parent, we have blood type O, which is designated by two eyes. At this point, you should have memorized the blood types, so combination AB, combination A, combination B, and then designation for O. It's just helpful when you're taking practice tests or questions related to college biology or AP biology. Okay, so now let's take a look at the Punnett square where you're going to have capital A and capital B designated for these, and then you're going to bring down the I's for each one that is listed here from the first parent up top. So we get these unique combinations. So what does this mean for the offspring? Well, that means that you have one set or 2 out of 4 that is going to be blood type B, and then 2 out of the 4 that's going to be blood type A. So 50% probability for blood type A and 50% for blood type B. So in this case, the recessive allele, hypothetically, is that little I. So unless there's 2 little I's, then phenotypically, you will not see the designation. So even though this is heterozygous and this is heterozygous, remember, it takes on the qualities of what's dominant, which is letter A and letter B, respectively. So hopefully that helps understand or help clear up some things. Now we're going to go into sex-linked traits. So sex-linked traits are the designation or understanding of sex-linked genes. For example, for females, remember you have 2 X chromosomes, and for males you have 1 X and 1 Y chromosome. What's unique about that is that males are more likely to inherit X-linked traits because it's really just all or none. We also call that hemizygous because let's say, for example, you have a female and they can carry a gene that is dominant for some reason, or they're associated with a dominant gene that has a disease. So let's say they're heterozygous. Okay, then they have the disease. Let's say they're homozygous recessive, then they don't have the disease. So it's either that or we have that combination. Well, think about this. It's only going to be one letter. So you either have A or little a. And so it's 50-50 for the male. Now let's reciprocate this and say that the disease is recessive. Okay, then the female doesn't have the disease, but then the male, again, has a 50% chance of having the disease. And we'll clear this up more in details in just a second, but common examples of this include color blindness and hemophilia. So let's take a look at an example problem. Take a moment, pause this video if you know how to conduct sex-linked trait pedigrees. If not, go ahead and pause this video and try to solve the following problem. Okay, so now that you're done reviewing the problem and potentially giving an answer, it says color blindness in humans is often due to a mutation in a gene in the X chromosome. Consider a mating scenario between a female who is a carrier of color blindness gene and a colorblind male. What is the expected genetic combination or outcome for their children? So they're asking for the genotype or the phenotype. So in this case, we're talking about color blindness, and color blindness is seen as a recessive gene. The reason why we know this is because it says there's a carrier of color blindness. So in this case, I just put a lowercase c for the female. You can put a uppercase C for the female. So basically you would have something like big C, little c, but because of the script and how C looks and how the lettering looks, I just put nothing for the female. And then since she's a carrier of color blindness, which is recessive, by the way, I put a little c there. So hopefully that helps clears up why I did that. Now for the males, you're going to have the male that has color blindness. He has the recessive gene. It's attached to the X-linked chromosome. Remember, Y is irrelevant in this case, so it's not going to carry anything anyway. And then you combo each one and you see that, remember, the ones that don't have a designation is not going to have color blindness. So who has color blindness? You have a female that potentially can be colorblind and a male that can be colorblind. So in sex-linked genes, you tend to divide it not by like out of four, but out of male and female. So we say for females, there is a 50% chance of essentially being colorblind and or being a carrier. And then for the male, we could say that the one male is unaffected because he does not have the color blindness gene. However, the other male does, and because it's a recessive gene, he'll take on that characteristics. So that's why characteristics such as color blindness and hemophilia are prominent in the male population. Okay, let's talk about incomplete dominance. So, incomplete dominance is a genetic phenomenon in which the heterozygous lettering is a blend of both parents. So, in this case, for example, if we were to take R and W, we take a look at this problem here. It's talking about flower colors and roses, and they let you know it's incomplete dominant. And they said red roses are going to be both capital R, and then white petals are white petal roses, I should say, is going to be both capital W. Well, what happens if there's a combination? Well, if there's a combination, that would be a blend. So if you have red and white and you mix that together, that'll make essentially a pink color. So they're asking for the potential pedigree of this. If you want to pause the video and try to complete based problem, go ahead. Once you're done, let's review the answer together. Okay, let's review the answer together. So in this case, we have one parent that has the red roses, so big R, and then the other parent that has the white petals, which is WW. And then if we have a combo, we have all RWs, which means that all offspring will be RW, which means the phenotype for all offspring will be that pink color. Now let's talk about codominance. So codominance is a little bit like incomplete dominance, where it's a combination for the heterozygous. However, in this case, the heterozygous would be a possibility of both showing up. And so, if, for example, if we were to say that the flowers scenario was codominant, then it wouldn't be a pink color. It would be like a red and white spotted flower if it was following codominance. So codominance essentially is a scenario in which both alleles are equally dominant. However, what's going to happen, instead of being a blend, like in complete dominance, it's going to be expressed equally if heterozygous. So take a moment and read the following problem. If you need to pause the video now, feel free to do so and try to create the pedigree or the Punnett square, and then we'll review the answer in just a second. Okay, so now that you're done completing that, you can take a look at the answer here. So they say a cattle rancher is studying color-coded genetics, and basically he notices that he has heterozygous cows. So you might see these cows outside in nature if you live in an area, and they're spotted. And if they're spotted like this, where they have a both combination of the black and white, they're designated as BW. And then if you combine them together, you have two big B's, BW, and WW. So what that means is, typically speaking, you'll have two big B's, then which is black. BW, you'll have two of them, which is the spotted ones, and then white, which is going to be WW. So that's going to be the combination of both. Now that we're finished focusing on those types of problems, something else to think about is non-nuclear inheritance. So non-nuclear inheritance is basically this idea that we get mitochondria. We talked about this in an earlier unit, which basically says that mitochondria has its own DNA. And we know this. And we know that mitochondria DNA is passed on by mother to child, not from paternal. And so in this case, sometimes you could have issues where there's inheritance or DNA that's passed on from the mitochondria. So just keep that in mind if you have a multiple choice question related to non-nuclear inheritance, which just basically means that we're talking about the mitochondria or the chloroplast, which has its own set of DNA that's going to be passed on from mother to child, and they're maternally inherited from that mitochondria. So it has its own, the organelle in the cell, the mitochondria, has its own DNA that will pass on from mother to child. So I hope that helps review some topics as it relates to the idea of several problems that you could see for the hybrid testing or for the AP biology exam. we reviewed a ton of different types of problems. Make sure to review those problems. And if you want to, please subscribe. Make sure to click the like button and also make sure to review earlier videos that we have posted as well for earlier units for review if you're planning to take the test anytime soon or tests related to AP biology or college biology. We hope you stick around for future videos. Thank you. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.